Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class we have talked about read mapping for transcriptome analysis and we have talked about some of the methods that we use. So, we will continue that discussion and we will talk about the next steps which is transcriptome assembly and quantification. So, these are the concepts that we will be covering today. So, the first concept is transcriptome assembly, right. We will talk about what is assembly, we will discuss some of the tools that are used for assembly and how do you evaluate the quality of assembly. And the second concept that we will be covering today is the quantification of transcript abundance, right. So, this is a very important step for any further analysis, right. So, we want to count, right, how many transcripts are there in the cell. Okay. So, this is what we do using this transcript abundance quantification. These are the keywords, one is assembly, another is abundance. Now, going back to the RNA sequencing data processing pipeline, right. So, we have seen this pipeline before. So, we start with the read data which is usually in FASTQ format if you are working with Illumina data especially. Then we do the quality control, this is common step, right, that we have also done for variant analysis etc. Then we have the mapping step which actually leads to this read mapping against a reference sequence. So, we have talked about three methods, we have mentioned three methods and we have discussed star and HiSet 2 and we have discussed HiSet 2 in much more detail in the last class. And once we have done the mapping, so this is done now. So, we can now do something called assembly. Okay. Also, we can do the assembly directly from the data itself. Okay. There are tools that will allow us to do that. So, this is something we will discuss today in this class as well as in the next class. Okay. And then once you have done the assembly, we can do the quantification. Okay. So, let us start with the transcriptome assembly. What do we understand by assembly? Okay. So, assembly means the reconstruction of complete set of full length transcripts from the read data. Okay. So, we have mapped the reads against the reference sequence, but these are fragments, right. So, you remember the steps, right. So, we have this RNA isolated, we have cDNA synthesis, we have fragmentation. So, the reads come from fragments of transcripts, right. So, in these cases what we want to do is we want to assemble the full length transcript from the fragments. Okay. So, this is what the assembly process does. Okay. So, we can use short and long read data, right. So, again depending on the platform that we are using, if we are using Illumina for example or Ion Torrent, we will get short read data, but if you are using SMRT or nanopore, right, we will get long read data, right. So, depending on the platform we can have different types of data. We can also have hybrid data, right. We can mix this short and long read data and we can do the assembly. Okay. So, the goal is to get the transcripts, but also to get the isoforms, right. So, we have discussed about isoforms when we are talking about the transcriptomic analysis, right, and we discussed how many types of isoforms you can get and what are the processes that can give rise to isoforms, right. So, this is something that is actually quite important to also get the isoforms, right, because we also want to know in many cases which isoforms are expressed, right, and maybe they have some specific function, a specific role in the biological process. Okay. So, this is summarizing what I just said, right. So, we have imagine we have a gene, right, with 3 exons, we have exon 1, exon 2 and exon 3 and present in the reference genome, and we have let us say short read data, right, where reads are mapping to these regions within exons, but also some reads might also map in this junction, okay, but that will be quite rare in case of short read data. But in case of long read data, right, in many cases you will see these reads that will be spanning these junction, right, these are splice junctions. So, you will get this kind of reads when you are working with long read data, right. So, one of the advantages of long read data is that you can then identify which isoforms are expressed, right, because of this presence of these junctions in your data. So, we can take this short read data and long read data and we can assemble the full length transcript. Okay. So, how do you do that? Okay. We will not go into a lot of details, right, about the algorithm, because the algorithm that is used is also common with the genome assembly process, and we will discuss about that algorithm in genome assembly part in much more detail. Okay. So, I will just mention the algorithm, some of the tools that are used for this process, but we will not get into the algorithm itself. Okay. So, what you get in transcriptome assembly is that you will get many fragments, right, that correspond to different transcripts, right. So, you have let us say 10,000 genes, you expect 10,000 transcripts, right. So, you will have these after the assembly process, you should get around that number, right. So, that is a quite a big number if you compare against genome assembly, right. So, if you have genome assembly, this is very similar, right. Transcriptome assembly is very similar to genome assembly. You will see when we discuss genome assembly process. So, in genome assembly, what you expect, right, ideally you expect, right, multiple chromosomes, right, assembled. So, if you have let us say 16 chromosomes, you expect 16 chromosomes, right, 16 fragments assembled, whereas in case of transcriptome assembly, right, you will expect maybe 10,000 fragments, right, again depending on the number of genes that are present in that organism. Okay. So, this is a major difference between transcriptome assembly and genome assembly. Right. So, this is in contrast to small number of large fragments, or what we call contigs in case of genome assembly. Now, transcriptome assembly it can be two different types. The first one is reference guided transcriptome assembly. So, as the name suggests, right, there is a reference genome, right, against which you are doing this assembly. So, we have genome of the organism or a closely related species that is used as a guide, okay, for the assembly process, or you can do de novo assembly, right. So, there is no reference genome and you have to make sense of the whole thing, right, somehow, and this is you can probably understand this is much more challenging, okay, where when you have a reference genome, this is actually slightly easier to do the assembly, whereas if you do not have any reference, this de novo assembly process is a much more challenging problem. We will come to this discussion later on when we talk about genome assembly, and I will show you, right, why this is a much more challenging process, and will illustrate, right, the algorithms, and you will understand why this requires a lot more computational power, a lot more complex algorithms. So, what are the major challenges for transcriptome assembly? So, the first challenge is that there is non-uniform coverage due to difference in expression and other factors. Okay. This is something that you probably realize, this is a difference, big difference compared to the genome assembly, right. So, in genome, right, you have just one copy of the genome, maybe some elements are repeated, right, or present in multiple copies in the genome, but they are not hugely variant, right. It is usually not the case that one region is present and 1000 copies, and the region is present in 5 copies. It is usually not like that when you are going for genome assembly. In transcriptome assembly, right, some genes they will be expressed at very high level. So, maybe there are a large number of RNA molecules are present in the cell, whereas some genes are expressed at very low level, maybe you have just 1 or 2 mRNA molecules present in the cell. So, given this difference, how do you actually assemble the whole thing, right? And this is even more challenging, right, for genes that are expressed at very low level, because you have probably only few reads mapping to those genes. Okay. So, this is one of the major challenge for assembly. In addition, what you get is that you also have non-uniform coverage along the length of a transcript. Okay. So, when a mRNA is produced, right, there is a cDNA synthesis step, there is amplification if you are going for Illumina, etc. So, all these processes lead to some sort of biases, right. So, we will talk about these biases a bit later in subsequent presentations, where we will have detailed discussion of these biases, but it will be enough to know now is that there is non-uniform coverage along the length of a transcript. So, if you consider a gene A, right, and you see the transcript A, you will not get reads across the whole transcript, right. You will see some bias, you will see some part of the transcript, especially for example, the ends of the transcript, they will show more reads compared to middle of the transcript. Okay. So, there are a lot of different types of biases that we will discuss in the subsequent classes. Okay. So, this is a major challenge again, right, given that you have some regions of the transcript have very high number of reads and other regions have low number reads, how do you actually assemble the full transcript? Okay, and the bigger question is how do you actually identify the isoforms? Okay, and this is the next point, right. So, isoforms are present for a transcript, right, and this could be present for many genes depending on the organism that you are working with, and what you have to do is have to identify which isoforms are being expressed. Okay. This is a challenge given short read data, because you will get very few reads that map across splice junctions, right. So, why I am focusing on these reads that map across splice junctions? Because these reads will tell us which exons are being connected, right. So, splice junctions will tell us whether there some exons are connected or whether some exons are skipped. Okay. So, these reads that map to the splice junction, these are very, very important for identification of isoforms. Whereas, in long read data this is slightly less challenging, right. You can get many reads that map across these splice junctions, and hence you can identify which isoforms are being expressed in the cell or in certain conditions, etc. On top of that, you have sequencing errors, right. So, again we have already talked about these sequencing errors, what type of errors you get in different platforms, right, and these will be there, and this is something that we will have to take into account when you are doing the transcriptome assembly. Okay. So, you now understand, right, the different challenges that are present when you want to do the transcriptome assembly, and we will now discuss some of the assembly tools and algorithms. So, when I discuss the tools and algorithms, I will just mention the tools and I will briefly mention the algorithms. We will not go into details of these algorithms, because it will not be possible to discuss all of them in short period of time. If you are interested, of course, have a look. I will give all the references in the presentation. So, please have a look into the algorithms, and I will just mostly highlight the algorithms and the tools that are used for these processes. So, let us start discussing these tools. So, many of these assembly algorithms or assemblers, right, these are the tools, use something called De Bruijn graph assembly approach. Okay. And this is an approach that is also used for genome assembly. Okay. So, I will not go into this De Bruijn graph assembly approach, because we will discuss this when you talk about genome assembly approaches, right. So, there are different approaches for genome assembly, but the predominant one that we use today is the De Bruijn graph assembly approach. We will talk about these algorithms, these approaches, their advantages, their limitations, etc. When we talk about genome assembly process. Okay. So, it will be enough to know that they use this De Bruijn graph approach, and this is a very efficient approach that works very well for genome assembly. So, this is for de novo assembly, and here are some tools that are available. Of course, there are many tools, many other tools available as well. So, one is Trinity, then it is a very popular one, then you have RNA SPA-DES, then you have SOAP de novo trans. Okay. So, again as I mentioned, I am just mentioning some of them, there are other tools out there. And as I mentioned, this is a really challenging process, the assembly of transcriptome, because you have many fragments involved, as well as the non-uniformity in coverage across transcriptome and across transcripts. Okay. So, this makes the whole process very challenging, and this is something you need to keep in mind. Okay. So, I will just mention the tools, and as I mentioned, these use this De Bruijn graph assembly. So, here is the paper describing the tool Trinity. Okay. So, it talks about the process of this assembly, etc., and you can also get the code in this link and the tool in this link, and if you want to use it, you can go there, download and install and use this tool. Okay. So, have a look. We are not going to go into the algorithm, as I mentioned, right. There are the algorithm is De Bruijn graph assembly, which we will discuss a bit later. Here is the next tool, which is RNA-SPA. So, this is again a de novo transcriptome assembler, and this is used for RNA-seq data transcriptome assembly. There is a equivalent tool which is called SPADES, right, SPADES, without the RNA part in the first, and that is actually used for genome assembly. So, as you see, right, these actually are kind of adaptation of genome assembly algorithms into transcriptome assembly. And we have another tool, as I mentioned, right. This is again a de novo transcriptome assembly with short reads. It's called de novo trans, and again you can go and have a look. So, we will discuss some other tools which also do assembly and as well as can quantify the transcript abundance. Okay. So, one such tool is string type, right. So, this is also helps in improve reconstruction of transcriptome, right. But compared to the previous ones, this is a tool that does assembly based on a reference. Okay. So, you get reference guided assembly. So, you have to have the reference genome present, and in addition to the assembly of the transcripts, this tool can also quantify the abundance of transcript that is present in the cell. So, what you will see, right, many of these tools that we discussed, they can do a lot of these different things and there might be some overlaps. So, it is not like strictly kind of defined, okay, this tool will do just quantification, this tool will do just assembly. There are also tools that actually can combine 2 tasks or 3 tasks together, as we will see as we go into this discussion. Okay. So, how does this tool work? Okay. So, I will mention this very briefly. As I have mentioned, this is reference guided assembly. So, you have the reference genome. Okay. So, when you have reference genome, you can have the alignment, right, reads will be aligned against the reference genome, and we know the alignment map, right. So, in the form of BAM file or SAM file, we know the alignment map. So, string type, it takes this alignment map and it builds something called a splice graph. Okay. So, what is this splice graph? So, in splice graph, the idea is actually very simple. You have, so it is a graph, means you have nodes and edges, right. So, if you are familiar with graph theory, so you will have edges and nodes. So, in case of a, so we will have these nodes like this, right, and we have these edges between nodes. Okay. So, these circles are actually nodes, and then you have these edges. Now, these edges can be directed or undirected. Okay. So, in case of directed, this is a directed graph, in case of undirected, this is an undirected graph. Okay. So, let us not go into a lot of details, but this will be enough, right. So, this node, there are nodes and edges. Okay. So, what these nodes represent in splice graph, they represent exons or part of exons. Okay. So, we have seen, if you remember this image that I showed you, we have this exon 1, exon 2, exon 3, right. So, if you have this exon 1 is one node, exon 2 is one node, exon 3 is one node, right. So, these nodes represent these exons or part of exons, and then you have these edges between these nodes. Okay. And if you find a read, right, so let us say it goes through a splice junction, you can have an edge between some of these nodes, right. So, for example, exon 1 to exon 3. Now, if you have these edge between exon 1 and exon 3, that means this is a splice variant that is present in your data. Okay. So, if you see reads, right, that show that map to the splice junction between exon 1 and exon 3, you will add an edge to the graph, and this will mean that this splice variant will be present, right. So, you can now imagine this in a much more complex example, right. Instead of just 3 exons, you can have 5 exons, 8 exons, and you have these connections, okay, and each connection represent a splice variant. Okay. So, maybe you can traverse through this splice graph, right, from exon 1 to exon 2, maybe to exon 5, then to exon 8. This would be a splice variant, right. So, where you have these exon 1, exon 2, exon 5 and exon 8, right. So, this is again kind of an isoform, right. So, this you can actually use this splice graph to actually identify which variants are present in your sample or in your data. Now, once you have built this splice graph, this method actually uses something called a maximal flow algorithm, right, which I will not discuss again, and it actually estimates the abundance of these transcripts. Okay. Each of these transcripts, right, there would be an abundance value that is estimated from this splice graph, okay, using something called a maximum flow algorithm. Now, string type can also quantify expression without doing any assembly, right. So, this is something you can say, okay, we do not do any assembly, just quantify, right, based on the data that is present, because we are giving the alignment map, So, you can simply say just quantify, and you will see there are tools that will not do assembly, they will simply go through mapping and then quantification, okay, without doing any assembly. So, string type can also consider annotation in GFF, GTF format. So, we have talked about this annotation right when we are talking about variant annotation. So, this is also important for transcriptome assembly, because you want to know, right, which gene contains how many reads. Okay. So, you need this annotation of genes or coding regions, etc. And it can also compare with known annotations, and it can generate statistics, etc. So, you can explore, right, this tool and see what are the features that that are available in this tool. So, again it is impossible to cover everything in the class. So, I will encourage you to go and check all the resources that are available, what are the options, the tools, etc., how do you run them, etc., all these things. Okay. So, the next step is, once you have done the assembly, right. So, what you want to do is you do something called quality control. Okay. This is something that is also done for genome assembly, and similarly we also have to do this for transcriptome assembly. Now, what do we check actually? What can you check that will tell us, okay, this is a good quality transcriptome assembly? Okay, similar to that we check in case of genome assembly. So, one of the things we check is something like sequence length and fragmentation pattern. Okay. So, in case of genome assembly, we have, let us say, in an organism we have, let us say, 16 chromosomes. Okay. So, what we want is that these assembled fragments, right, they should be around 16 in ideal scenario, right. So, we have complete assembly, we get 16 fragments, 16 contigs, that represents 16 chromosomes. Okay. So, similarly here we have an idea about the transcripts, right. We are working with an organism where we know probably around 10,000 genes are expressed, let us say. And if we see that, like, there is a huge number, right, much more than 10,000 fragments that are obtained after assembly, this might mean that the assembly process was not very good, right, or there was not enough data, right, for the assembler to actually do the complete assembly. So, this is what we can assess, right. So, if you have too many very short fragments, so this could be an indication that the assembly was not very successful. Okay, and this can be due to sequence quality, or there could be some issue with the assembly process itself, because of some peculiarity of the data, the assembly process did not work very well. And another measure that we can also check is the fraction of reads that is used in the assembly. Okay. So, if the assembler used the most of the reads, that is actually a good sign, right. So, assembler could utilize most of the data for the assembly process, right. So, that is why if you have like a most like higher utilization, that is a good sign that the assembly actually worked very well. Okay. So, the next step, once you have done the assembly, you have kind of evaluated the assembly quality, the next step is to do the quantification of transcript abundance. Okay. So, how do you actually quantify the transcript abundance? Okay. So, this is the process where you actually convert this mapping data to abundance data, and this process can sometimes go through the transcript assembly process, as you have seen. What we get after this step is something called the raw count data. Okay. And this is a very important step in estimating and comparing gene expression level. Okay. This is a very important step, and you will see, if you look at the databases, you will see a lot of data is deposited in terms of raw count data, because this is the starting step, starting point for lot of downstream analysis. Okay. So, we will talk about again some of the tools, at least one tool which we use for this kind of analysis, and of course, there are many tools that we will again discuss in the next class. So, one of the tools that is very popular is called cufflinks. Okay. And this is for, again, it can do transcript assembly, and then it can do the abundance, and as well as it can do something called differential gene expression analysis. So, we will talk about differential gene expression analysis later. So, as you can see, this can do a lot of these things together. Okay. Again, we will not go into lot of details. Here is the link to the paper, and also the link to the tool. You can actually explore and see all the details of what it can do. So, very briefly, what it does is something called, it builds something called an overlap graph from read data. And how do you build that overlap graph? Again, remember the graph theory, you have nodes and edges, and here reads that show sequence overlap are connected by edges, right. So, the idea is the reads that show overlap, they are probably coming from the same transcript, right. They are part of the same transcript because they are showing sequence overlap, right. At least part of them are identical in sequence. Okay. So, that is why they are presented in graph. Again, this is a very similar idea, again we will illustrate a bit more when we talk about genome assembly. Now, what it does next is it actually identifies the minimum path that needs to be traversed to generate transcripts. Okay. Again, it is a graph traversal algorithm, and then followed by that there is something called a maximum likelihood abundance estimation from the graph itself. Okay. So, again we are not going into all the statistical details what is maximum likelihood abundance estimate. I will just mention this is what it does. In case of you are interested, please have a look how this maximum likelihood method work. This is a method that is widely used in statistics and in lot of applications. So, very briefly here are some functions that you will find in couplings. For example, couplings, cup merge, they are used for transcript assembly. You have also the tool for differential expression analysis. You also have different other visualization tools, etc. Again, I urge you to explore through the website. Now, what are the challenges for transcript abundance quantification, right? So, we will end with some of these discussions, right. So, one of the things is that many reads do not span over the splice junctions, right. So, as we have seen in case of short read data, they are mostly mapping to the exon regions, right. Most of them will be mapping to the exon regions. So, then using these reads, right, in case of short read sequencing, how can we assign these to isoforms? Okay. How do you identify isoforms, right? This is a huge challenge for us. And how do you actually assign gene expression? Do you say average isoform expression or overall gene expression, right? This is something again could be interesting depending on the research question that you have. Okay. For example, you want to identify which isoforms are expressed or whether some isoforms show higher expression in certain conditions or certain treatment, etc. So, in those cases you want to do the isoform expression, right, and not just the overall gene expression. And also again, as I said, you want to look at differences in isoform expression across experiments. Okay. So, I will just mention some of the tools that we used for transcript abundance. So, couplings is one such tool, again it does a lot of this assembly then quantification of abundance, right, and this is again very similar to string type that we just discussed. There are dedicated tools now that actually will do this transcript abundance estimation, right. And from alignment map we have something called HTSEC count, feature counts, we will discuss them in the next class. Couplings we have just mentioned, and on top of that you also have alignment free methods. Okay. Again we will discuss them in the next class what these methods are. Here are the references that we have used for this class, and to conclude, so we have talked about transcriptome assembly, and this is a challenging problem due to several issues that we have mentioned, right. We have non-uniform coverage, we have isoforms, we also have sequencing errors, etc. We talked about two types of assembly. One is de novo assembly, right, where there is no reference sequence, and then we have the reference guided assembly. And as we have seen, right, de novo assembly is much more challenging than the reference guided assembly, and we have discussed some of the tools that can do de novo assembly, right. We have not discussed algorithms, which we will discuss later when we talk about genome assembly. And then we have talked about the transcript abundance quantification, and this is a very important step in gene expression analysis where we actually get the raw count data, and as I mentioned, this is the data that we will see when you look at gene expression data across different studies. Okay. This is the data file that will be available for most data sets, okay, because this is the starting point for all downstream analysis. And finally, we have talked about some of the tools for assembly and quantification, right. We briefly touched upon them, and I of course encourage you to explore more on these topics. Thank you.